Okay, my name is Dishetha Mamidi. I go to Ed W. Clark High School in Las Vegas, Nevada. I'm a junior, and my research paper is about the production of different types of recombinant antibodies as cancer treatments. So in my introduction, my area of investigation was recombinant antibodies, which are basically like a type of monoclonal antibodies, but they're made using recombinant technology. And my research question was how do the production methods for different formats of recombinant antibodies compare to each other? And my objectives were to find similarities and differences between the different formats and the productions, since there are various different formats, like structures, for how recombinant antibodies are made. And so in my literature review, my two main things I wanted to focus on was like how monoclonal antibodies were made and like the different structures of recombinant antibodies. So for the monoclonal antibodies, they're made using antibodies from one's own cell line to create like lab-grown antibodies. And historically, they've been produced using hybrid donor technology. That's the most common method, where basically you take someone's like antibody-producing cells from their B cell line not someone's, but like some organisms, and then you combine that with an immortal myeloma cell, and that'll make a long-lasting monoclonal antibody that can target a specific antigen. But the problem with this is, like I mentioned, they come from like organism, usually like mice, which creates some problems in like being compatible with humans. So as of recent, recombinant technology has developed, and it's been found to be more efficient in various ways. I wanted to focus on monoclonal antibodies using recombinant technology. And there are various formats of this type of antibody. SCFE, which is single chain variable fragment, FAB, which is fragment antigen binding, and BSAB, which is bispecific. As you can see in this one, the first image, like there's the monoclonal antibody, that's its structure, and then the fragment antigen binding and the SCFE are basically like parts of the monoclonal antibody, but like isolated. And down here it's not labeled, but it's a bispecific antibody. And as you can see, like the two top variable parts of it are different, so they target different epitopes, they are different epitopes, which means they can target two different antigens or like two separate parts of the same antigen. And my methods was basically literature review. I didn't analyze any data or do anything hands-on, since I was just comparing the different productions using like sources I found. So in my results, I talked about the production of fragment antigen binding antibodies, and there's three main steps. Like that, to simplify this image, there's three main steps: derivation, selection, and expression, where you have to get the DNA put into a host cell to make recombinant DNA, and then you select the DNA that will that's the most specific when targeting antigens, and then you use the recombinant DNA put it into a host cell line to actually make the antibodies to express them. And I found that SCFE antibody production is very similar. There's just a few differences, such that in FAB production, at least in the specific type that I analyzed, usually you use E. coli as the host cell. But the problem with that is you need to make sure the expression of the DNA is there's soluble expression for the antibodies to actually be formed. So to ensure soluble expression, there's two different methods between FAB and SCFB. And then here in this image, I also talked about BSAB production, bispecific antibody production. And there are various different formats of bispecific antibodies, since they're made by combining two different types of antibody formats. So I decided to just focus on the ones used combining two SCFE formats. And here are the examples of various types of these scFv tandem BSABs. And they're all just made by making the scFvs and then combining them with using some type of protein linker. So in conclusion, to answer my question of how do the different production methods of different formats of recombinant antibodies compare to each other, I found that scFv and FAB productions are very similar, and BSAB production varies the most due to its amount of different formats. But there is still a lot of overlap between BSAB, scFvs, and FABs by default because BSABs use like two different formats, and so you have to actually make those formats first before you combine them. So you actually have so that part of the production overlaps. I faced various limitations, such as like I had a lack of access to papers behind a paywall, so that made it harder for me to find research like different productions and understand them better. There was also I also had minimal mention of different functions of each format, and I did not analyze any experimental data, which could have like added new findings to my study. There was like a surprising lack of clinical studies, which I believe is because scFv and FAB antibodies, many of they haven't. There's none that are like FDA approved. I think there's a few that FAB ones that are FDA approved, but they don't target cancer. All the scFv ones I found that were approved were bispecific actually, so they're the most common type of bispecific antibodies. But yeah, it made it harder for me to find clinical studies. That's it. Thank you. How do I stop sharing?
Great job, Dishetha. You can stop sharing. Do you want to share your experience with Gifted Gapper?
Yeah, I would like to thank the two professors, Professor Prahlad and Professor Virgil. They were really helpful in Professor Prahlad's lectures in the beginning of the program. That's how I found my research topic of monoclonal antibodies and narrowed it down to get into recombinant antibodies through some research. And I think both the professors are really helpful in helping me like format my paper, figuring out how I was going to format it, and giving me information like fixing my grammar and stuff. And also, specifically, Professor Virgil was able to help me with a lot of parenthetical citations that I had a lot of questions on when like using the APA style. So I think it was overall a really positive experience.
Excellent, excellent. Professor Prahlad, a question for Dishetha.
Yeah, so, Dishetha, excellent, great talk, great research. So you talked about different types of antibodies. So FABs or bispecific antibodies or single chain. So which one do you think would you pick if you had to pick one for like application in cancer?
I feel like using BSABs, the bispecific ones, is probably the best, just because out of all of them, like BSABs are the only ones that have been FDA approved for cancer so far, and they also combine different types of formats, so they are able to target more antigens or like better target a specific antigen, which makes them more effective and it's and like targeting tumors and stuff. Yeah.
Okay. All right. Yeah. Thank you. Great job. Congratulations.
Great job, Dishetha.